Hello everyone, I'm Hongxing Shi from Nanjing GenScript ProBio. Today it's my pleasure to give you a webinar on the topic Robust Stable Cell Line Platform for Biologics Development. I'm going to talk about today. First, I will have an introduction of cell line development, and then I will introduce the workflow of cell line development and give some case studies from ProBio. And last, I will give an introduction about GenScript ProBio. Cell line development plays a key role in the development of biologics. Cell line screening is the initial stage of CMC study. All process development work needs to rely on the development process of cell lines. The quality performance in the cell line development stage will determine the difficulty of process development and the development strategy. A stable cell line with satisfied yield not only makes the development process efficiently, but also helps reduce the cost of the entire production. Quickly obtaining stable and high-expressing cell lines is a direction that every scientist pursues in cell line development. There are two questions for us to find the answer. One is, how do we get high-expressing cell lines, and what determines? For this question, from our experience, it's determined by the candidate molecule and expression system. The second question is how to efficiently and quickly screen high-expressing stable cell lines. That is, how to consider and design screening methods and evaluation strategies from the perspective of technology and workload. Cell line development refers to the process of introducing foreign genes into host cells and isolating them through screening techniques to obtain positive clones. Based on this definition, host cell line, vector system, and clone selection technologies constitute the three essential elements for constructing cell lines. Another important aspect is how to use these three elements to achieve cell line construction. This is the second question we just mentioned, how to do in process control, how to design the entire screening process and the clone evaluation strategy. The whole process needs to be well controlled to ensure that the stage results of the final. Such in-process control is a decisive factor for determining whether a cell line development platform is robust enough and flexible enough. Although this term is actually used to describe such as cell culture process and purification process, it's also very appropriate for cell line development. The robustness of the cell line development lies in its ability to ensure the quality of cell line, and the flexibility lies in the fact that the development process can be adjusted according to the changes in the needs of the project. And it will not affect the main line of cell line development. Robust can be used to describe the timeline, the stability, and the productivity of the selected cell lines. For flexibility, for example, in the face of different molecular structures, additional design needs to be carried out on the basis of platform solutions, and the special attention should be paid to some risk points. These concerns can be reflected in the plan. For example, in the process of project development, there will need for providing materials quickly for the purpose of process development or quality analysis. This need can occur in the transfection stage, in the pool screening stage, or the clone screening stage. Another aspect of flexibility is that through the experience database, the results can be prejudged in advance within the controllable risk range, thereby accelerating the development. Next, let's talk about the key elements of cell line development. For the host cell in the starting technology, there are very clear requirements for it. First, there is clear historical background information which can be traced well. Second, the relevant tests have been carried out, including identification, sterility, mycoplasma, viruses, etc. Third, it can be well adapted to serum-free suspension culture to meet the need for scale-up production. Fourth, it can secrete exogenous proteins well and have good post-translational modification. Fifth, it can maintain stable production capacity and stable product quality. And moreover, it can perform stably in multiple projects and help form a platform. Chinese hamster oval cells are the most popular used host cells, and 80% of antibody drugs are produced by CHO cells. This picture is a traceability picture of CHO cells. It can be seen that CHO cells were first isolated in the lab in 1957, and a variety of commercial cells have been generated. Among them, CHO-K1 cells that have not been genetically modified are stored in ATCC and ECACC. Both cell lines are adherent culture and need to end serum. In 1973, CHO cells that can be used for suspension culture were isolated and named CHO-S cells. In the 1980s, a single allele knockout cell, DXP11 of DHFR, appeared. In 1983, CHO-DG44 with DHFR biallelic knockout appeared. This cell has no DHFR gene activity and can better perform its screening function. Similar to the DHFR gene is the use of GS gene. The GS gene knockout was CHO-K1SV GSKO from Lonza. They are also chosen cells from Merck. For the key elements of the starting technology, the vector generally includes a promoter for the expression of the target gene, poly-A, restriction sites, and the screening genes. Whether the design of the vector is reasonable, especially the design of the target gene expression cassette elements, will affect the yield of the cell line and the ratio of high-expressing clones, which will affect the efficiency of screening. The integration site and the transcription efficiency of the target gene in the host cell will affect the performance of the clone. How to reduce the influence of random sites on the expression? Strategies that can be used, such as adding elements that can increase transcription activity, or you can choose to target the gene integrate into the active site of transcription. You can also use a strong promoter to drive the target genes and use the principle of co-amplification to express the target gene, such as the DHFR and GS we just mentioned. After talking about host cells and vectors, let's talk about screening techniques. Here are several common screening methods, including manual operation and equipment use. They are limiting dilution method, FACS, ClonePix, VIPS, and the Beacon platform. Among the screening technology, the limiting dilution method is easy to apply for screening, and there's almost no damage to the cells. But in order to obtain enough clones, both the plating density and the medium need to be optimized. FACS needs to be fluorescently labeled. ClonePix needs to use semi-solid medium, and the method for judging the high expression of secreted antibodies needs to be optimized to improve the accuracy of the judgment of high productivity clones. VIPS is equipment that can replace manual plating operations and needs to be combined with an imager to record monoclonal pictures. Beacon is a fully automated, which can shorten the time for clone screening and evaluation, and the process has image data. In the process of cell line screening, monoclonality assurance is a keyword. The acceptable strategy has several options. One is to carry out 2 rounds of low-density limited dilution, which has certain requirements for the density of plating. For those using FACS and clone peaks, the correct process and parameters can be used to increase the probability of obtaining a high percentage of clones. It's also acceptable. In addition, it can be used with images captured with an imager taking pictures at the beginning of after plating. Here are clone images that are taken at different times after cells are distributed into 96 well plates. There are 2 pictures under the wheel at each time point. One is a picture of the whole well, and the other is a magnified picture of the cell location. The first image is on the day of plating, we can see 1 cell in the well, and the second image is 24 hours later, there are 2 cells. Left image is 48 hours later, and there are 4 cells in the well. The right is a picture of the cells on the 3rd day after plating. These are pictures of the cells on the 7th and 15th days after plating. If there are 2 cells on the day of plating and the cell will not selected, or in the following days, the number of cells will lead to misunderstanding. For example, if the number of divisions is not regular, it will be excluded. The above is an introduction to the 3 starting technologies involved in cell line development. Next is the introduction of cell line development platform of GenScript ProBio, including the workflow in process control and the case studies. In this picture, according to the commonly used strategy and different stages, cell line development can continuously supply materials to other modules. The cell pool can be provided for scale-up culture so that materials can be provided in time for the process team, as well as for developability research and in vivo study. In the clone stage, a platform evaluation program is used to select better clones as soon as possible so as to provide research cell bank PCBs for process development and analysis method study. In the MCB WCB stage, cells are provided for pilot production. All these tasks put forward requirements for cell line development, and a robust cell line development platform needs to be established. Next, I will share with you the cell line development platform ProCellD from ProBio. As shown on this page, the package of ProCellD covers DNA synthesis to the completion of the PCB constructions of 6 preferred clones. These studies can be completed in 40 weeks. Our in-process control first is to divide the process into 4 stages. First is DNA synthesis and plasmid preparation. The second is the stage of pool screening and selection, which includes screening of transfected cells under pressure. And at the same time, the platform solution is used to evaluate the pool so that the better pools are selected to enter the 3rd stage. The 3rd stage is single clone screening and selection. In this stage, cells are plated into 96 well plates and images are captured. Preliminary evaluation using the platform solution will be used to select 6 clones for fourth stage, which is PCB generation. During the process, our focus is in process control. The control points shall specify the amount of work to be completed and the goals to be achieved at each stage. The workload and the target setting of the control points should be quantifiable and easy to measure. Ensure that each stage is operated on a reasonable scale and can achieve reasonable results. These figures are finalized based on the data and the experience of the platform. In the cell pool screening stage, what needs to be controlled is the total number of cell pools obtained and the cell pool expression level range. Only after these 2 points have reached the standards can we consider that our process executed correctly and the screening results are reliable. For the clone screening stage, also, what needs to be controlled is the total number of monoclonal clones obtained in the screening process, the distribution of clone expression levels, and whether the monoclonality images meet the requirement. At this stage, for clone evaluation, what needs to be controlled is the growth profile, expression level, and the key quality indicators of each clone meet the requirements. And for risk control, we need to set up a backup plan from a technical point of view. The in-process control is to set up different stage for the entire process, which is based on technical considerations and is convenient for drawing conclusions and making plans. The second step is to set goals for each stage, that is, set the goals of the cell line module based on the overall target product attributes. For example, the desired expression level is 3 grams per liter. What is the goal for each stage so that we can increase the probability of final delivery? The third step requires a scientific workload to ensure that the data is enough to move forward. The fourth step is to conduct risk analysis and set up backup plans to reduce the uncertainty in the process. The setting of these indicators should be quantified as much as possible to facilitate implementation and judgment. This picture is a general introduction to the ProBio cell line platform. This platform has more than 12 years of experience. The cells we use are Choke 1, after thermal-free suspension culture. The traceability is clear. The platform is equipped with automated equipment and multiple imagers to meet the needs of monoclonality. We are constantly optimizing the process, and currently we can complete the delivery of DNA synthesis to top 6 clone PCB within 14 weeks, and the introduction of Amber 50 mini bioreactor combined with our robust process strategy can complete the top 1 clone selection within 18 weeks. At the present, the antibody production can reach 3 grams per liter and up to 8 grams per liter. It has excellent stability performance. In many projects, the 60 to 90 generation has excellent stability performance. The ProCLD cell line platform has delivered more than 300 cell lines, of which more than 80 cell lines are used for IND filing. 6 cell lines have entered the clinical research phase. 5 are in clinical phase 1 and 1 in clinical phase 2. Let me share with you a few specific cases. Here are the performance of cell lines in 3 different projects and different stages. The first case is the expression level of pool is 0.7 2 grams per liter after batch culture evaluation. It reached the level of 3 grams per liter in the cloning stage after 14-day fed batch culture. In the 90th generation stability study, the yield was maintained at 75%. As for the second project, the pool can reach 4.1 grams per liter after 12 days of fed batch culture. The clones reached the level of 4.2 in 14-day fed batch culture, and the stability is maintained at 89% for 60 days. The third case is the bispecific antibody. The pool can reach the level of 2.3 grams after 12 days of fed batch culture. The clones can reach the level of 3 grams per liter for 14 days fed batch culture. The stability is maintained 98% after 16 days. All the fed batch culture has used our platform strategy, which has been verified by multiple projects. It can improve the efficiency of clone evaluation and save time. And at the same time, this data can also reflect that the good correlation between pool expression and clone expression and a reasonable prediction of clone yield can be made in the pool stage. Here is an analysis of our current production level for MAB. It shows that the average production can reach 4.35 grams per liter, and at the same time, the platform's experience in non-MAB molecules is gradually accumulating. Everyone is very concerned about the diversity of clones, hoping to screen clones from different single-cell sources through different transfections and different pools. The ProCLD platform is also very concerned about this. Here are the preferred 6 clones from 2 pools of different transfection batches. The ProCLD cell line development platform can be well connected to the cell culture platform. What is shown here is that the cell line has the similar performance from the small scale to large scale, and the growth performance, expression level, and charge variance can show good scalability and stability. We just mentioned that the last stage of the cell line life cycle is MCB-WCB production. After the successful development of a cell line, the top clones will enter the cell bank production. ProBio has the ability to conduct bank production under cGMP conditions, which is our GMP Pro cell banking service. Is an introduction to the ProBio cell line development platform. Next, I will spend some time introducing our GenScript ProBio. We wish to become your most reliable and sustainable biologics partner. Our mission is to dedicate it to proactivity, providing end-to-end service from discovery to commercialization with professional solutions and efficient processes to accelerate drug development for customers. Business footprint. In 2004, we established antibody discovery capability. In 2008, we established the capability of CLD and process development. In 2017, we assisted a customer to obtain their first IND approval from NMPA. In 2019, we officially established the Biologics CDMO segment and launched the BioGMP R&D Center and the GMP Virus Manufacturing Center. In 2020, we launched independent GenScript ProBio brand. Our new cGMP facility for clinical production was in operation in October. Our business includes antibody drug development and gene cell therapy. We aim to provide one-stop CDMO services from discovery, preclinical development, early clinical, late-stage clinical, and commercialization. This is our core competencies. For Bio, as a one-stop CDMO partner, we have experienced people and the project management team, as well as innovative win-win business model. That is all I'm going to talk about today. Thank you for your listening.